experiment is designed to study the genetics of cancer. The study will link the appearance of cancer to specific gene abnormalities. Which of the following forms of cell molecular analysis is most useful to identify gene alterations involved in carcinogenesis? Immunohistochemistry, single nucleotide polymorphisms, flow cytometry, fluorescence in situ hybridization, and tumor marker expression. So what do you guys think? Yeah, D. Huh. B, it's fluorescence in situ hybridization. You remember last time we talked about fish and we said it's used to localize DNA or RNA sequences in a sample. Use it to find the location, the presence, and the location of specific gene abnormalities, basically. If we go to immunohistochemistry, what does it mean? And it's in the name, immunohistochemistry. Can anyone try? Okay, to save time, immunohistochemistry, it basically uses antibodies to detect the location of proteins and other antigens in tissue sections. That's the thing, it's in the name, immunology and histology. So use antibodies to detect the location of antigens in tissue sections. Then flow cytometry, we know it deals with identification of cells based on their properties like surface proteins and cell size, those kind of properties. So that's not a helpful answer here. And then tumor marker expression. Tumor markers, okay, remember we were told tumor markers only there to monitor like malignancy progression. And they're basically secreted by cancer cells or other cells in the body in response to a tumor, basically. So the molecules, just a minute. So the molecules that are secreted by cancer cells or other tissues in response to benign conditions or cancers. So yeah, in this question, the best answer is fluorescence in situ hybridization. We go to number 90. So, I want to minimize this thing. The best method for detecting a clonal T process is by cytologic examination for nuclear irregularity, DNA gene rearrangement studies, immunohistochemistry for T cell markers, electron microscopic inspection of nuclear features, in situ hybridization for T cell DNA. Answers, guys. What do you guys think? EC. What else? So for this question, at first I was going E, in situ hybridization for T cell DNA. But then I saw that the best answer Because remember, what is a clonal T cell process? What is a clonal T cell process? What does that mean? Anyone who knows? Like, you type it out really fast so that we move fast. Yeah, it's you remember in the formation of a T cell, you had that process, yeah. Okay, clonal T cell process is basically having T cell clones, T cells that have a similar BCR receptor. So you remember when we were forming a T cell, there was that part, yeah, VDJ recombination, and that's what was being rearranged so that you get different T cells. So I think that at that point, you need to study that point so that you can tell whether they're clone T cells if you look at that recombination. And that's why I was going with B, DNA gene rearrangement studies. And I actually found an article that was supporting that answer. Like, that's the best method that's what they usually use to detect clonal T cell processes, through checking that point, yeah, VDJ recombination. I don't know it like to detail. I can't remember honestly, but you guys know it's there. At that point as well, there's a lot of recombination, changing it, just recombining it to create different T cells. So yeah, I was going with B. Someone who actually typed it here, but I can't remember. We go and that study, that DNA gene rearrangement studies via PCR. Then we go to 95. So an immunoglobulin causing a paraprotein can be identified through serum electrophoresis of urine, blood protein electrophoresis, immunofixation, quantification of kappa and lambda chains, vanillyl mandelic acid analysis. So what do you guys think? Someone else? So I'll send the article. See, yeah, immunofixation. So, first of all, we ask ourselves, what's a paraprotein? I went with C. What's a paraprotein? A paraprotein is an immunoglobulin fraction that are produced of B cell lineages. So basically, it's immunoglobulin fractions produced by clonal population of B cells. Nini. You know, B cell lineages can produce clone cells, clone B cells, and those clone B cells will produce Igs. So those are the paraproteins. So choice A and B, serum electrophoresis of urine and blood protein electrophoresis. Those two tests are usually used in checking paraproteinemia, like checking if those immunoglobulin fractions are many, their presence, and how, if there are many. But this question is asking us to identify a particular paraprotein. So we'll use immunofixation, because what is immunofixation? It allows detection and typing of monoclonal antibodies or immunoglobulins in serum or urine. So in order to answer this type of questions, I feel like you need to know the definition of all these terms. So that will help you to answer the question correctly, because the question is asking an immunoglobulin causing a paraprotein can be identified through. So here you're looking for the immunoglobulin, not checking paraproteinemia. That's why I went with C. And then something else to note, immunofixation uses precipitation as a method of analysis. Then we go to 99. A 2-month-old male presents with persistent diarrhea, signs and symptoms of pneumocystis jirovecii, pneumonia, and an oral fungal infection with Candida albicans. His weight is in the 10th percentile. Test results for HIV are negative by PCR. The most likely cause of these findings is, so we need to highlight the most important things. There's diarrhea, signs of infection from pneumocystis, and Candida albicans infection, fungal infections. So in answering this question, we need to know like what fights fungal infections, which lymphocyte or which immunoglobulin, whatever, which lymphocyte usually fights fungal infections, which one is the most common in fighting fungal infections. You don't even have to tell me like the major one, just B cell, T cell, inini. Try, and then also, yeah, T cells. T cells are used to fight fungal infections. So here, we know that fungal infections in newborns are mainly due to T cell defects. So what disease are we thinking about? T cell defects. Yeah, DiGeorge syndrome. So the answer here is defective T cell function. So we're not thinking about B cell disease because the child has immunity from the mother for the first few months of life, which will cover the bacterial and viral infections, etc. But fungal infections usually covered by T cells. That's why we're considering it as a defective T cell function and not these other choices that are associated with B cell disease. And then also note the child is a 2-month-old. So the 2-month-old is still using the mother's immunity. Yeah. Then we go to 103. Yeah, and if you haven't understood anything, just tell me to repeat. We'll go back to it. Then, which one of the following listed substances is secreted by malignant plasma cells in individuals with multiple myeloma and is the osteoclast activating factor that produces the characteristic lytic bone lesions? So, what is multiple myeloma? Before we even look at the choices. Just the summary of that definition. What does it involve? No, before even we go to that, yeah, it involves plasma cells. So, yeah. So it involves abnormal plasma cells. And we need to know what those abnormal plasma cells usually secrete. Can you guys give examples? So the question is, abnormal plasma cells usually secrete? Question mark. Yeah, thank you. Tumor necrosis factor alpha. Yes, interleukin 1, rank L. Yeah, those are enough. Even 3, interleukin 3, I saw it somewhere. So here, the answer is interleukin 1, and it's also an osteoclast activating factor. So remember those three are important. TNF alpha, interleukin 1, and rank L. If you need to note that down, it's in the chat box. So next we go to 111. So, a pediatric patient with a history of recurrent infections is suspected to have a defect in the neutrophil-mediated killing of phagocytosed bacteria. Which of the following lab assays is most suitable to confirm this suspicion? Quantitative serum immunoglobulins, complement fixation test, complement blood count and differential, nitroblue tetrazolium dye reduction test, flow cytometry for CD18 expression. So, what disease are you thinking about even before we start? So, history of recurrent infections suspected to have a defect in the neutrophil-mediated killing of phagocytosed bacteria. So, which disease is that? I said in that previous question, Needy George syndrome. And we said interleukin 1 because multiple myeloma is a plasma cell discrease, that word, whatever. It secretes abnormal plasma cells, and abnormal plasma cells usually secrete TNF alpha, interleukin 1, and rank L. So we went with interleukin 1 because it was the one that was in the choices. Yeah, the answer is D, nitroblue tetrazolium dye reduction test. So for this question, yeah, I asked which disease, it's CGD. Chronic what? Can't remember the name. It's actually down here. Granulomatous disease. Yes. Which other name is? Which other disease is usually associated with neutrophils? You guys need to know these things. Those diseases. We have, let me give you the examples and then I'll ask you guys what it entails. So we have CGD, chronic granulomatous disease. That one we've already said, it's in the chat box. It's a defect in the NADPH oxidase. So no reactive oxygen species can be formed in order to have efficient phagocytosis, basically. So yes, the pathogen will be phagocytosed, but it won't be broken down. It won't be killed. Higher. We also have leukocyte adhesion deficiency. What does that entail? What's the problem there? Leukocyte adhesion deficiency. Yeah, impaired migration and chemotaxis of neutrophils. Jeddy syndrome. Yeah, so many. I remember there were under those notes for phagocytosis, phagocyte defects, sorry. Okay, So we said, by the way, we're still in that question, yes. So for this question, let's go back to our question. We said this is probably because it's a defect in neutrophil-mediated killing of phagocytosed bacteria. It's probably chronic granulomatous disease, where there's a defect in the NADPH oxidase that produces those reactive oxygen species. So the phagocyte will phagocytose that bacteria, but since hakuna ROS there, you won't be able to kill that pathogen. So we can use all these tests from A to D. We can use them, but there's one that is the most suitable, which is D, as someone said, nitroblue tetrazolium dye reduction test. And how it works is that deceased neutrophils, now the neutrophils that are not producing that NADPH oxidase, are unable to reduce oxidized nitroblue tetrazolium to insoluble blue formazan. So that's the principle of the test. So those, and you can see like the test is saying it's a reduction test, so it's unable to reduce that nitroblue tetrazolium. That's the whole principle. Now this flow cytometry for CD8 expression, I feel like because leukocyte adhesion deficiency, now that disease it also involves like impaired neutrophil leukocyte chemotaxis and migration. It's a CD18 integrin defect. So I feel like flow cytometry will be best for assessing that leukocyte adhesion deficiency. Flow cytometry for CD18 expression. Now we go to the next question. After we've already answered it. Defects in neutrophil NADPH. Yeah, defects in neutrophil NADPH oxidase system produce streptococcal infection, leukocyte adhesion deficiency, celiac Higashi disease, and Hashimoto's disease, chronic granulomatous disease. We already said the answer is E, chronic granulomatous disease. Hashimoto's disease, that's an autoimmune disease of the thyroid. I already explained it in the last session we had. Then celiac Higashi disease, we've said it's also defective. It's a defect in the neutrophils, basically. There's neutrophil dysfunction. And it affects intracellular protein transport. Then leukocyte adhesion deficiency, I've already told you guys it's a CD18 defect. So basically what happens is that adhesion molecules that allow phagocytes to adhere to the vascular endothelium, adhesion molecules usually allow the phagocytes to adhere to the endothelium and migrate till they pass and go to the infection site. But in this defect, there'll be the adhesion molecules are deficient, so they'll be impaired chemotaxis, like that movement will be impaired. They won't be allowed to adhere to the vascular endothelium. I hope I'm making sense. Let me just check there. Yeah. Okay, so we go to wow. We go to 134. This is also a question that is direct. The following statements regarding cytokines is false and true. They're small proteins, 5 to 25 kilodaltons. They act through the cell surface receptors. Some may enhance or inhibit the action of other cytokines. produced only by immune cells. A given cytokine may be produced by more than one type of cell. So what do you guys think? Yeah, the answer is D, produced only by immune cells. So that's not true. Which other cells can produce cytokines? Guys, give examples. Yes, hematopoietic cells. Which other cells? Fibroblasts can also produce that. Cytokines. Endothelial cells can produce cytokines. Okay, I'll give you three. Fibroblasts, endothelial cells, hematopoietic cells, immune cells. Those are enough. Then we go to 144. So which of the following lab immunological techniques can be used to detect maternal anti-rhesus factor against the baby's rhesus factor in a suspected case of hemolytic disease of the newborn? I'm even doing this question, but okay. So we've been told to detect maternal anti-rhesus factor against the baby's rhesus factor. So this is basically a Coombs test, but you want to know if it's the direct or the indirect one. So Coombs test, basically you're trying to detect antibodies against an antigen, right? But where the difference between direct and indirect is location of those antibodies. So direct uses antibodies, the Coombs reagents. It uses that to detect presence of antibodies or complement on the surface of a cell, of a red blood cell. So you have presence of antibodies or complement on the surface of a red blood cell. But indirect is the presence of antibodies in the serum of the mother. And it doesn't detect complement. That's also a difference. So direct is on the surface of the red blood cell, but presence of antibodies in the serum of the mother is indirect. So like here, the question is, can be used to detect maternal anti-rhesus factor against the baby's rhesus factor. So maternal, meaning the serum. So answer is E, indirect Coombs test. Then 147, okay, yeah. The fluidic system of a flow cytometer is designed to deliver cells in an orderly single file stream to the interrogation point so that only one cell passes through at a time through a process called, what do you guys think? The fluidic system of a flow cytometer is designed to deliver cells in an orderly single file stream to the interrogation point so that only one cell passes through at a time through a process called. Yeah, answer is hydrodynamic focusing. Okay, you guys know how a flow cytometer looks. So, in order to get, like, if you use common sense, even without knowing anything about a flow cytometer. In order for the cells to pass in a single line, common sense will entail you to make that cut tube really small so that those cells pass one at a time, right? But we don't have any tube that is that small because cells are so tiny. You can't create a tube that is that small. So we use the power of fluid dynamics. So hydrodynamics uses fluid dynamics to make sure those cells pass through one at a time. So what you do is you have the sheath fluid. If you know part of the flow cytometer, I feel like we can discuss flow cytometry really fast. Let me see where my notes are. Basically just the components of a flow cytometer. So components, we have the fluidics. Fluidics meaning the isotonic sheath fluid that moves the sample stream. So that key fluid that's outside that fluid in a tube. The tube that is usually used in a flow cytometer is just a normal tube, but then you put a fluid inside, and that fluid is called the sheath fluid. Then you're supposed to inject the cells, but they're suspended in a fluid, in another fluid. So those cells, it's like when you inject water, oil in water, they're going to mix, but they'll create a kind of laminar flow, because actually you can't choose that choice D, laminar flow, because that's the optimal condition for the sheath fluid. It's not the principle that is. That's just the condition for the sheath fluid, so that it doesn't mix with the cell suspension. So we said you have that tube, you have the sheath fluid, the normal fluid that's going through that tube, and then you inject the cells. The cells are suspended in a fluid solution. So since they don't mix, the cells will be passing one at a time. Other components, we have the laser, we have the optical bench. Those are photodetectors that convert light into electric signals. We have the electronics that convert analog light signals to digital light signals, or basically info that is processed on the computer. Then a computer to analyze that data. Yeah, so I feel like that's enough for now. Something else you need to remember, like sample preparation, we use whole blood and it's stored in EDTA. That's one thing I remember we were told to note. And the sample is not supposed to be clotted, and definitely because you want those cells to pass through one at a time. So no clotting is allowed. That's why we're using EDTA. Yeah, I feel like that's okay. I hope you guys have understood how it works. Then choice number 148. So you say the answer is E, hydrodynamic focusing. (148) Opsonization of bacteria occurs through coating bacteria just with. Remember, we discussed this last time and we said, what is opsonization? This is basically coating cells with opsonins that will change the structure, or molecules. Let's use molecules, and those molecules are called opsonins. So coating cells with opsonins that will alter the surface of that pathogen such that they'll be able to be detected better. So opsonins are molecules that attach to the foreign target cell and alter the surface of the cell in order to ease phagocytosis. Like, it makes the whole process more efficient. So opsonins, what are the opsonins? Remember, I told you guys it's IgG. Which segment? The FC portion, not the Fab. So choice E is out, not Fab, it's FC. Then we also have C3b. Kwanza C3b is the one even in the textbooks. They have a whole diagram about it. But basically, it's the fragments of the complement pathway that are recognized by type 1 complement receptors, so CR1. And those fragments of the complement pathway that can be recognized by CR1 are C3b and C4b. But mainly we use C3b. That's why we've gone with choice A. So IgG, Fc portion, C3b, C4b. Those are the optionings. Then number 150. Plasma protein electrophoresis in a neonate with emphysema or recurrent respiratory tract infections may show a deficiency in the following protein. So this one, I just went with pathology. Emphysema, you remember the pathophysiology of emphysema? There's something to do with tobacco smoke, and the other one was, what's the answer, guys? Yeah, that one was the mechanism of, that was based on alpha-1 antitrypsin deficiency, where if you have alpha-1 antitrypsin deficiency, it was something to do with protease, antiprotease. You guys will just go through that. Honestly, can't remember it to detail. But I remember, macrophages, when they phagocytose cells. See, in the lungs, we have, whenever you have a pathogen, the macrophages will work to kill that pathogen. So as they're killing, they're producing, the proteases will be activated. They either produce proteases or proteases will be activated. And then now your body is supposed to produce antiproteases so that they deal with those proteases. So basically it's like, what do you call it? Like you create a problem and then your body produces a solution, like something to fix that problem. So a feedback mechanism, yeah. So basically, if you have that alpha-1 antitrypsin deficiency, now you won't be able to produce, you won't have antiproteases to control the proteases, so you'll have like that damage the alveoli. So yeah, the answer is D. I was basically just explaining the alpha-1 antitrypsin deficiency and its relation to emphysema, but answer is D. So yeah, we're done with the MCQs.